Perfect. Okay, I think that people are still joining, but I think we'll go ahead and get started because we want to make sure that we have our full hour. So if I can just remind everybody that, say we're on a Zoom call, if they could please just turn off their cameras, and everybody should be muted for right now. And then we'll take calls, we'll take questions at the end. And please, you can type them into the chat, or you can also ask questions at the end, and you can raise your hand. And that, the way to do that would be ready to turn your camera on. So I can think of no more qualified speaker to talk to us today about SARS-CoV-2 than our speaker, Vaithilingaraja. Vaiti actually trained as a veterinarian at Madras Veterinary College in India, but then he moved to the US to do his graduate studies in cell and molecular biology at the University of Arkansas, followed by a postdoctoral fellowship here at UCLA, where he studied virology and stem cell biology with Ren Sun. After a short time at Cedars, where he ran the virology core, we were very lucky to recruit him back to UCLA. And his lab is focused on understanding the mechanisms of RNA virus replication and host pathogen interactions. And he's published extensively on hepatitis C virus and Zika virus, and he's a CERM-funded investigator on Zika virus and brain organoids. However, when the COVID-19 pandemic started, he realized that he could use his BSL-3 training and his knowledge of RNA viruses to advance our understanding of SARS-CoV-2 and to find new therapies. And so he's become one of the busiest people on campus right now. He's spending countless hours in the high containment facility while collaborating with numerous groups to answer specific questions about the virus. And we're really glad that he could find the time to speak with us today. I also just want to say that on a personal note, Vaiti is an absolutely wonderful collaborator. He's always ready to help and to share his insight and his ideas for experiments, no matter how busy or how tired he is. Even if it's 2 o'clock in the morning, he's there to help. He's also a really wonderful mentor and teacher to our students, and we're just really fortunate to have him here at UCLA. So I could go on and on, but I don't want to take up any more of his time, because we're really looking forward to his talk, which is titled Cell Models and Tools for COVID-19 Antiviral Drug Discovery.
Thank you, Bridget. So today's talk, essentially, I want to focus more on the resources we have generated in the past 6 months and how this can be utilized, and also some of the projects which are already mature and they are in revision with journals. And also we are, as Bridget said, we are collaborating with the multiple groups. We learned a lot in the short last 6 months period about this virus. So essentially the title is Cell Models and Tools for COVID-19 Antiviral Drug Discovery. So this is the stat today. We have about 13.5 million confirmed cases and over 500,000 deaths. So it is a pretty serious disease, and it kind of flip-flopped everybody's life. I think parents, grandparents, and professionals. I think it's the whole humanity is in a crisis mode. So if you just take USA alone, there is 3.5 million cases. And in Los Angeles, 140,000 cases. Just mind-boggling. And also in our hometown, close to 4,000 deaths already happened in the last 4 months. So when in January, mid-January, they were talking about this disease in Wuhan, China. At that time, we read like about 400 patients with 8 deaths. So so far, I've been working with Zika virus since 2016 outbreak. With Zika virus, we see the virus affecting the pregnant woman, causing fetal abnormalities. But there's only one case of death, and there are estimated several million people, but the death was very rare. So here, when there was 8 deaths, about 400 people infected in mid-January, it's kind of ringing bell for us. So by the time third week of January, the cases were started coming up. The numbers were coming up, and at that time we want to learn more about this virus. So at that time, right now the virus name is SARS-CoV-2, but at that time there's no name, and they were using novel coronavirus 2019. So we were actually going through the sequences, and we want to know how this virus evolved in a period of time, and whether we could be able to develop vaccine. So we pulled together the data, and we posted in the preprint. At that time, if you look at the newspaper, this virus outbreak happening somewhere else, we are so it's not close to us. So there are a lot of people want to learn about this virus, but there's not much information there. So this was one of the top trending articles in that month. So just to highlight few of our descriptions in that paper. So we were mid-January, we had one sequence published around January 7th or 10th. But by January 21st and 27th, we got about 40 genome sequences coming from China. So we pulled them together. The one thing striking was this virus, very actually close to the bat than the human. This virus is very close to the bat than the previous SARS or MERS coronavirus. So that is one thing confirmed by other groups as well. So here, another important question. This question we were trying to address at that time, but now it is kind of everybody's asking how this virus came, and a lot of controversy surrounding that. However, the one thing clear is this virus about 96% similarity to the bat coronavirus. So the source should be more likely it's coming from the bat. This bat coronavirus sequence was described in 2013. So there are probably a lot of back and forth transmission between bat and human happened, and that's likely contributed to the current outbreak. With the original SARS virus, it has 78% similarity when you compare the whole genome. So here is the timeline where you could see in 2002, 2003, the SARS-CoV outbreak, the original one. Okay, there are about 8,000 people affected. About 774 people died. Similarly, the 2012 and 14, the MERS-CoV, about 2,500 people affected, 866 dead. But however, if you look at our current, this SARS-CoV-2, there are millions of people affected. So even though it says 13.5 million cases, CDC estimates it's a tenfold higher, because several of the countries, they don't have a high number of diagnostics done. And also there are a lot of asymptomatic cases. So whatever the numbers we have is essentially kind of an underestimation. So here I get back to the question, viral evolution, viral origin and evolution. If you look at the SARS-CoV and MERS-CoV, it's actually when it was infecting the human, the human body doesn't know how to react. The same way the virus doesn't know how to react. So there's a lot of confusion. And at the end, you just get a messy relationship. The virus almost kills the host. So if you take a very perfect pathogen or a parasite, they try to establish a balance that they don't want to kill the host. They just want to coexist so they can survive. But the SARS-CoV, the old SARS-CoV and MERS-CoV, they are just kind of confused viruses, and our body also confused. So there was a lot of fire, so we could easily detect those viruses or the cases, and we quickly control them. However, this virus, the SARS-CoV-2, is a very smooth operator. I was telling my colleague, this is almost a well-designed Ferrari. So it is so beautiful, it can transmit so efficiently. If you have one person infected in a crowd of 200 or 300 or 1,000, you will going to have a 80–90% transmission rate. So if somebody says this is coming from an animal, this virus should be very confused, just like the SARS or MERS-CoV. However, the SARS-CoV-2 is so smooth, it's already established a kind of a best equilibrium. That's why we have this pandemic. So that's just to make that point. So when all these things are happening in the end of January, we actually reached out to UCLA biosafety team, especially Sam Chow. I was discussing with him, this is what's happening. What do you think? He was very supportive of this research. However, I was very nervous whether we should take a plunge or we pull back. So when I had a couple of inflection points in my mind. Number one, SARS-CoV, if this pandemic virus, this virus has only less than 8,000 cases and less than 700 deaths, there's no point in doing this. This is probably another SARS virus. Okay. But when by February 1st week, already China had like a 60,000 cases with over 1,000 or 1,500 mortalities. So it's very striking. Okay, this is important. This is something we should pursue. So then we talked to the IBC, our UCLA IBC, they were okay. So at that time, we didn't have a viral strain to work with. So essentially, we wanted to synthesize the genome and reverse engineer the virus. So within 10 days, we got the viral genome synthesized. And we still have this genome available. It's in a multiple fragments, so it's not infectious. However, we give out these genome fragments to other investigators at UCLA. So they had a head start to clone multiple genes, and those projects are ongoing right now. While we were discussing the possibility of using live virus at UCLA, on February 7th, I just sent an email to, they recommended me to contact Barbara Dillon, our high containment director. So I think it was the best move I made because when such a sensitive virus outbreak coming, people are very cautious not to get involved. So that is the case in other UC campuses. However, here UCLA, she was so helpful, and she said, "Okay, let's. What else you need?" And also, she was very mentor-like. She was advising, "These are the things need to be done. You had to get it done." So we worked very closely for the next few weeks, and there's no viral strain available. So by the time March 1st week, the CDC actually deposited the first virus isolated from January 21st, they isolated from Washington. The first patient came to USA. So that's the strain, and they made it available through BEI resources. So we worked with the BEI, essentially the ATCC, and we got the virus strain on March 13th. So we made a stock of this virus, multiple stocks, and we established a lot of assay systems that I will focus in the next 5 minutes. Essentially, this virus we received March 13. We made a stock by March 17th. Then we measured the virus titer. The CDC recommended using TCID50. These are all the ways how to quantify the viral particles, infectious viral particles in a culture. We tested different ways. One is the standard plaque assay with overlay, agar overlay. That's very low throughput and very tedious. So we gave up on it. And also you could actually fix and stain. So a lot of things went on in that quick period of time. So we figured out the viral titer, everything. Within the next 10 days, we set up the a high throughput drug screen. So this was another interesting phase we were in. The bulk of that talk I'll present in the last section of this presentation. But essentially, I reached out to Robert that he made that happen. So by March 13th to March 30th, we actually completed the virus production, drug screening, and all the assays completed. So it was very, very super fast. And also, we were the first campus, UC campus, to get this virological assays and virus set up. I would say even the whole West Coast. So we were ahead of the other team. So that gave us a head start. So now we have this virus stock available for UCLA researchers who has the approval to work in BSL-3 facility. Only thing, you just acknowledge the BEI for using this reagent, okay? And also, if you have any question for viral culturing and all the assays, let me know, or you can talk to Barbara as well. So here, so the panel A here shows we were using the monkey kidney epithelial cells. It's a vero cells. When you add the virus, here what you see is mark means uninfected cells. Is very clean and nice. But by 48 hours, you will get a lot of CPE. Means the cells are, the infected cells are dying and started floating away. So that's the CPE you could measure. So at that time, this all happening the mid-March, we didn't know what antibody to use. So we got several antibodies from BEI. They were used for the SARS-CoV, the original SARS. And quickly we narrowed down to about 6 antibodies. From that, we frequently use the guinea pig antibody and also the 240 monoclonal antibody. The guinea pig antibody is also working well in Western blotting if you want to use it. And also, we have used these antibodies as a neutralization platform. It's very interestingly, the guinea pig antibody and the 240C both are working. Both are blocking the virus so in a neutralization assay. So if anybody wants to use viral neutralization with antibody-based, you can reach out to me. And also, we were using the double-stranded antibody, double-stranded RNA antibody. So essentially, we have these resources, and also nowadays, many of the commercial vendors started providing antibodies as well from mouse or rabbit. Okay, if you need, I can provide that. And here, just to show the uninfected cells and the infected cells where the spike protein is localized. Right? The spike protein is the key for viral infection. So another thing is the qPCR to measure the viral genome. This is critical as well to quantify virus replication in a cell or in a serum or in a plasma sample or tissue samples. So we have a very good control, and we can go down to 1 copy. And with water, you get about 35 CD cycles. It gives you a very nice R value. We also have the system available for researchers to use it. So during this time, we know the monkey kidney cells are working very well. But you know, a lot of interest from pharma and also us. We want to have a human cell lines or cell models to study this virus. So at that time, we tested multiple lung cell cultures. The first one we tried A549. People already knew this is a resistant line, but this cell line is expressing high amount of ACE2. So there's another layer of blockage happening, but that's a resistant line. Anything on the red is resistant line. Human primary fibroblasts, there's a little bit of infection, very low efficiency, but got shut down. And another thing, we tested about 7 bronchial epithelium immortalized cells and also the primary cells. The issue is they don't support the virus that efficiently. So we kind of at one phase we were very, very frustrated because we didn't have any system to work with. But another, there are 2 other cell types you could use. Is the 293T is a human kidney epithelial cells overexpressing ACE2. From UCLA, Koki actually made this cell line, and we tested it's working really well. And also this cell line also available through BEI. Another cell line is the CALU3. It's a small cell lung cancer line. It's also a very good cell line. So anything on the green is good to go if you need to use for your assays. But while we were struggling with these lung epithelial cell lines, that's the time we had a kind of organic collaboration with Bridget. So she had this air liquid interface culture developed from human basal stem cell of the proximal airway. So this is the concept is simple. You essentially grow these cells on the surface where the liquid and air interacts. So most of the cell outside apical surface are exposed to the media. So here you could see this proximal airway. It's a kind of a pseudo stratified layer with the basal cells, acetylated tubulin ciliated cells. So here you could see the ciliated cells are really infected well with the SARS-CoV-2. The green is the SARS-CoV-2. The reds are the cilia. So when we saw this, we were on the cloud 9 because we by first week of April we had a system to work with. So we were very happy. I had a video here. It's just gone, but I'll keep going with the next slide. Then we also looking at other cell types in the ALI culture. Here is the muc cells, mucin producing cells. That's also very well infected. So last week, Bridget gave a talk the previous week, 2 weeks back, and she covered all these details. And we are very fortunate to work with her group on this system. So we also have the system, and if any of you wants to use this ALI culture, please talk to Bridget. And also here, another interesting finding, when the infected cells, the basal cell type, this is more like a stem cell, but during infection, it is kind of very stiff and highly expressed, and also there looks like some proliferation. So there is a lot going on in the proximal airway system. So this can be used. So with that, I will stop the proximal system. So another project we've been working on from the beginning is using the lung alveolar 3D organoid culture. This is a complex system because the stem cells are from human lung, cryopreserved lung tissue, or coming from the discarded lung cannot be used for transplantation. So Barry Lab at Cedars-Sinai, they have established all the tools and reagents to generate these organoid system. So essentially, here in the panel A is workflow of how this whole system is done, but I'll just go through very quickly. So the distal airway organoid is like this. There's a lumen in the middle, and outside are the cells. Okay. So in the beginning, very beginning, we were just using this organoid as such, and assuming when you add the virus to the organoid, the virus can get inside and infect the cells. But these are polarized cells, so the ACE2 is actually expressed in the middle of the lumen of the alveolar organoid. So in the beginning, we were just adding virus outside, nothing happened. So this is what you see. It's a kind of 3 weeks of work. For Barry's lab, it's probably 45 days of work to get these organoids ready. So it was another phase. At that time, nothing was working. Then what we did, we took these organoids, we kind of pipetted them up and down and to disrupt a little bit. So there will be some space for the virus to get inside the lumen. Then things started working. So it's another eureka moment where we actually able to do the lung organoid system working. So this is very important because we see the other top labs in the USA, they are still struggling to get this system working, but we got it working, and this is a paper already in the preprint. If you have a chance, take a look. What you see here in the red is the type 2 cells, and they are the organoids and fully infected by the SARS virus. And also during viral infection, you could see the green. That's cleaved caspase apoptosis. So the cells are undergoing apoptosis. So once we have this system, we actually wanted to use this for drug studies. That's what Barry and we did. Here is essentially the RNA-seq data. It's a good system to study drug, antiviral drugs. And you could see here we tested interferon beta, that's blocking the virus, and also hydroxychloroquine is working as well. And also the remdesivir. Here, at least the remdesivir is a winner. We have done multiple times, at least three different donors triplicate. It's very intensively performed experiment, very rigorously done. So we have this system for pathogenesis study and also antiviral testing. So that's a kind of a lung story. When we were writing proposals, I think for CERM in April 7th, we had a lot of data we had accumulated. But the thing is, we were writing this virus affects the lung. It's a respiratory virus. But by mid-April or late April, we started seeing patients having underlying heart disease are highly susceptible to this virus. So that's a kind of a different take than the regular SARS-CoV. So this is another avenue we want to check in. But the heart here, this heart can be directly infected by the SARS-CoV-2 or due to hypoxia or the inflammation caused by the virus can damage the heart. So it could be direct or indirect. So the myocardium, we just were using the iPSC technology. So my laboratory been using iPSC for studying the viral diseases. So we were using iPSC-derived hepatocytes for hepatitis C virus in early 2010s. We were using iPSC-derived neurons and also retinal cups, optic cups for studying the Zika virus. So it was a kind of a natural transition for us to utilize this iPSC cardiomyocytes. So at that time, Clive Swenson, my former director in the RMI, Sinai, he actually, his postdoc, very active. He was keep emailing, oh, can you test these cells? Can you test these? But we were at that time mainly focused on the lung cells. But we got the cells from Arun Sharma. So this work was done very, very quickly. We had all the assays worked out. So essentially what you see here is the, the virus can actively replicate these iPSC-derived cardiomyocytes. You could see the red. And also, we could see like a holes in the monolayer. Here and there, you could see holes. So the virus is actively replicating, also ripping apart the cardiac muscle cells. So this was very striking, and we actually looked into the virus replication compartments in this cell type. So while we submitted this to the big journal, the main question they asked, is that a real infection? Can you prove it's a real infection? So we were using ACE2 receptor antibody. So when we use that antibody, the replication is completely blocked. So the virus is actually going through the ACE2 receptor. So that's one thing we confirmed. The other thing we worked out is when we do different doses, even 0.001, extremely low amount of virus, by the time day 3, they can overtake this culture. So this is one of the highly sensitive cell types we've been using. And when we did the RNA-seq, you could see 70% of the 50 to 70% of the RNA in the cellular transcriptome is from the virus. So this is another good system for us to work out. So here, essentially, the quantification of various markers we tested, the infected culture. Mostly the SARS is in the infected system, and also the double-stranded RNA, apoptotic cells, and all the co-infected cells. So when we were doing this experiment, we essentially taking the video of each time point. This is day 3. So when we were taking with the infection, you could see the heartbeat is there. It's kind of amazing to see the cells growing in the culture as can beat. So, however, with the infection, there was a striking phenotype. It is actually the video is going, but there's no beat. So here we quantified this. So the take-home message with this system is there could be a theoretical possibility the cardiac myocytes can be infected with the virus, but still, we have to learn whether is it really happening in the human body. But there is evidence the virus RNA actually present in the cardiomyocytes. So that's a good system to use besides the lung epithelium. This work is published. So here, this is, I think, the last section of the talk. Essentially, this is all happening in March. Once we have all the cell system worked out, I kind of emailed Robert on a Friday evening, and I said, can we do some drug screen? Mainly, we were focusing on some kind of nucleoside analog that can, because we know nucleoside analogs for HIV, HCV, or any of the viruses, they are key antiviral compounds. So he was suggested, why don't you use kinase inhibitor library, because most of them are nucleoside analogs, but with a still very specific activity towards the kinases. So we got excited. So we went through in a formal 48-hour discussion. By Friday, we had a discussion, and Monday we set up the screen. We kind of used about 430 compounds with various controls. So that was very quick. It's a one-day work. Then we did the infection in our BSL-3 facility. After 48 hours, we did the imaging of viral infected cells. So we essentially scored if there is cell death or viral CPE, that drug is not working because it's not blocking. If the cells are alive and you see a monolayer like MOC, uninfected cells, then the drug is working. So that was a very binary decision. So we quickly narrowed down to 34 kinases. So by Friday, Monday we started the drug screen. By Friday we narrowed down to 34 kinases. But the important thing is we got this list, and I sat down with Robert on a Saturday afternoon. We were social distancing, so he was sitting like a 10 meter away from me, and also I'm working with BSL-3 agent, so people are a little bit maintaining not 6 feet, 10 feet distance. So it was interesting to see. So I was telling him the compound names. He was like, oh. So by the time he said, this is very critical because it's all these compounds, 34, are clustering around a small set of kinases. So this is essentially the screen. So so here is the interactome map. You can see the panel B is the interactome map, where you could see about 20 of the compounds clustering around the mTOR AKT pathway, PI3 mTOR AKT pathway. This was very, very striking. And also a few of them are actually around ABL BCR genes, ABL BCR. That's a nilotinib. So we were very, very excited to get this data because people have done previous protein-protein interaction studies with the SARS-CoV, not this SARS, the other SARS, and they found out a number of viral factors are actually interacting with this mTOR pathway. So we got really excited because if the hits are all over the place, then there's not much to do. So this was an exciting phase. So within this all happening March 30th. So April 7th, April 6th, we finished the secondary screen. Here we had a discussion with Don Cohn, Dr. Owen Whitty, and also Bridget, Robert. We just kind of discussed a little bit of compound selection. So finally, we selected a compound which is not in the cluster because we know people will go after this cluster, but it is a little bit outside, but it is very good in the efficiency and also efficacy. So here this is a secondary screen with various doses. The key is you could see vehicle alone, no infection. The red is the SARS-CoV-2. Here you could see the with this COVID-2 with the vehicle, you see a lot of virus replication. But with the Berzoserti, we do see it's an inhibition. It's a dose-dependent inhibition. And also nilotinib there, but it's very good at 500. Then it was kind of not that active. But there are also other number of compounds that we could fall back on. Okay. So here is the IC50 curve we calculated. You could see Berzo is around 4 nanomole. So they all in the nanomole, sub-micromole range. That's very good. The one thing we found is the mTOR pathway compounds are a bit toxic. If you have anything above 25 nanomole or 100 nanomole, it's very toxic to cells. So the mTOR pathway, the compound range is 2 nanomole, 5 nanomole. It's a very narrow range to the toxicity and the therapeutic. But Berzoserti, we had all these up to this. We tested the viability is very good, 100% viability on these. So we need to integrate that data to here. But however, this allowed us to select the compound Berzoserti. The compound is owned by Merck. So we actually, Merck is very happy to support this project. So we are getting clinical grade compound from them for our subsequent studies. The important thing is in the second half of the talk, I spoke about the iPSC derived cardiomyocytes. So here we use that system for verifying the Berzoserti activity. So this is very good. Here we included hydroxychloroquine in the second experiment. Second and third experiment we used remdesivir. So it's very consistent. The Berzoserti is working very well. So it's blocking the virus and also reducing the apoptosis caused by the virus. So here it is a little bit of the zoomed in view where we without drug treatment like this and with drug treatment, you have a very good block. So with the Berzoserti, it's more like our this is a heartbeat. Anyway, essentially with Berzo, with the treatment and SARS-CoV-2, it actually keeps the beating going. That's the key thing. With the SARS-CoV alone, that's a small twitching here. If you see the video, small twitching. Let me see here. This video. Anyway, you could see the measurement here. These videos are already actually posted in the bioRxiv preprint if you want to look through. So we have completed these experiments and we are now focusing on more on the mechanistic side. Berzoserti is a ATR kinase inhibitor, so we are actually doing more on the pathway analysis. We kind of missing this slide from here. But essentially, this slide, we put all the pathways, the drug compounds blocking, and also that has an effect on virus replication. And also we had a Western blot analysis of the ATR DNA response pathway in this slide, but somehow this didn't come out very well. So I think I'm kind of in the last few minutes. The future perspective, this is kind of a very tough time for everybody. My kids are not going to school, so we have to find a way to, balance our life. And we thought the virus level is going down, so we can open up the businesses, economy, but the virus actually waiting for those kind of opportunities. So I think we are in a kind of a sticky situation right now. When in April, I was thinking maybe by October or November we will get 50 million cases globally and about 1 million deaths. So that was I was kind of based on the trend at that time. But last month I was very optimistic. The cases were going down. It's a very good feeling. But now we are back again. So which is telling, and people had theory with summer months, the virus will go away. No, it didn't go away. And also I do know tropical countries like India or Iran, all the countries, and also even in summer in Australia, didn't reduce the viral burden. So as long as the virus can able to find a warm-blooded body, it's going to jump in, whether it's a winter or it's a hot summer. However, my worst fear is coming winter. So we're going to have a double blow because the SARS-CoV-2 is going to be around and also the flu is around. So that's going to be a very tough time to go through. So we need to kind of stay safe. The one thing, this is more of my perspective, my opinion. So when I grew up in a tropical country, India, I actually, by high school, I had twice malaria including the cerebral form. The cerebral form is worse, so you go through that shivering and also the uncontrolled convulsion. Then I had typhoid, I had chickenpox, I had tuberculosis. So by the time you are a teenager, you are kind of bombarded with these diseases. But here, when I moved here for my graduate school, it's a very different perspective on infectious disease. So it's not a very important aspect to focus on. But that is understandable. However, the times are changing. The mosquitoes, they need to be in the tropical right now in Los Angeles. They actually can spread Zika virus, chikungunya, dengue, all the tropical diseases. So we are in a kind of a very, very changing world, and also these viruses are constantly evolving, and cities like New York are high dense cities. The virus going to exploit. So we really need to take a step back and think a little more futuristic. So I feel UCLA is a leader in the world, and also has a huge responsibility for the public health. So we currently working in a BSL-3 facility, which is very small, compact. We have about 60%, 65% capacity now. But by last month, I thought the virus will go away and we will have reduced capacity, but it is not. So we're going to have research pickup, and also we start getting more grants in. So we will have active research. So another thing, we have done these drug screen, we got the drug. What to do next? There are only 1 or 2 animal BSL-3 facilities in the East Coast. And when we reach out to them, they are already have a lot of on their plate. So we cannot do any more animal studies. And I do know other major institutions, they already put their papers in the preprint servers to show they have animal facility, animal studies. So I think moving forward with this context of infectious diseases, we do need to build up our capacity. Animal BSL-3 will take another 3 years or 2 years to build. At least if the leadership kind enough to listen and invest on this, we'll be really in a better situation the next time. And also another thing I noticed, I've been bombarded by infectious diseases during my college year, high school years. So I always aware of the infectious disease, how much damage it can cause. So from my perspective, usually when a virus outbreak is not there, they are actually preparing for the next attack. It might take 6 years, it might take 10 years, but believe me, they are preparing for a next outbreak. So there will be more coming. So we need to be prepared. That's just my humble request to our leadership. And with that, I think I'm actually running out of my time. Here I want to thank, this is just a 2-person race so far. The hero of this race is Gustavo Garcia, our research associate in my lab. So you will be seeing more publications coming in the next several months. Anything it touches with virus, virological assays, we both did it. So I really want to thank him, and also all the wonderful collaborators who've been supporting us. Sometimes we call them midnight 1 o'clock. Sometimes we had meetings on a Saturdays or Friday night, so they were there. So that's the energy we take it. So here I have to thank Robert. He was actually the pillar of that drug screen project. Bridget, we have a very strong, very, very successful collaboration so far. Don Cohen, Sam French, Koki for the antibody blocking. Melody, she's a wonderful colleague. Anton, we have very interesting project evolving, but he did the microscopic analysis. Orion, And also I have a special thank to Steve. He gave my cell phone so I can talk to him anytime. And also my colleagues at UCLA, Sinai. And also very important is our high containment facility director, Barbara Dillon. Without her foresight and strong leadership, I don't think we would be in this good position studying this virus. We already learned a lot about this virus. And also thanks for the funding to our School of Medicine, OCRC. I'm really thankful to the committee. Thank you, appreciate it.
Thank you so much, Vaiti, for a wonderful talk. I apologize, we had some technical issues because of some bandwidth, so some of the audio went in and out, but I think that a lot of people probably have questions. So I don't see anything in the chat, but if you have a question, please raise your hand or turn your video on and we can start. I think Sang Lee maybe wants to start.
Yeah, great. Okay, thank you very much, Vaiti, and for sharing so many exciting results. Can you hear me?
Yes.
Yeah. So I have a question about the drug discovery. So now people are looking for effective therapies that many different approaches. For example, the drug can inhibit the entry of the virus into the cells. Some drugs can inhibit the replication of virus. And here you are trying to inhibit the interaction between virus and cells and help improve cell functions. So what is the future of this drug discovery approach, and what kind of a drug will be likely to be effective for clinical applications?
So here it's a great question. Actually, we've been debating as well. So if you take the SARS-CoV-2, in the phase 1 of the infection, the first 10 days you have asymptomatic. So I think the virus is going there. It's actually replicating. And at this time, if you use a direct acting antiviral, can specifically target the viral protease or viral RNA polymerase, that's a good start. But you know, the body also changing, right? By the time you are getting into the day 11 or day 12, you have a lot of inflammation. Already a lot of signaling pathways are completely in disarray. So at that time, you really need a combination of therapy. So our approach is still valid. So it's only the finding the window where we can get the drug to the patients.
Thank you.
Can I ask a question?
Yes, please, go ahead.
Yeah, so Vaiti, I really appreciate that you're leading the way, getting the method and the research set up for all of us can use. So you mentioned the several systems that include the viral cells and the HEK cells, and the organoid. So which system do you think would be the best for drug discovery purpose to start with?
I think the issue we are dealing with here is when you have the complex system like the iPSC cardiomyocytes or organoids, it takes 30 or 40 days to get the cells to certain stage. So those are all the systems you can use for validation. But for drug screening, I think you still need to use Vero E6 or Calu3. You know, that's
I see. And for the hit that you mentioned, when you find the hit, do they work in all these systems when you validate, or there are some that work?
Because we from the beginning these are the drugs we used are from cancer, right? For cancer. So if you use them in a cancer cells like Calu3, the toxicity is very different. Some of the compounds have a different phenotype. So that's we kind of from the beginning we understood that issue. So we may use the 293A cells and cardiomyocytes, and now we are planning to do the lung epithelium. So you have to kind of choose.
I see. Thank you.
Can I ask a question, Vaiti?
Go ahead.
Vaiti, it's Moshe Arditi. How are you?
Hi, Moshe. How are you?
Great lecture. I know we talked this, but do you have capacity of having access to mutant virus if we have shown, for example, one mutant may do different pathogenesis or entry or infectivity? Is that available? Are they available, these mutants?
So we look through, the only source we can get is through the CDC and the BEI. Those are the natural mutants. Of course, the virus started drifting, so there are additional mutations in the genome. But we went through the catalog and we aligned the sequence. They are all same. So even though they say the strain came from Seattle or came from different places, they still have the same mutation. So we need to, that's one thing. It's essentially the availability. So if they are available, we can use it. And also, if they are from clinical sample, we can use it.
Now, for example, I know Cedars has been doing some genetic studies. They're about to publish this. I'm sure UCLA is doing. For example, you can have a hypothesis that asymptomatic carriers may be infected with a different type of virus as opposed to those who, you can ask a lot of good questions if that's possible to do.
Yes, if they are available, we will be very glad to have them. Thank you.
Hi, Veti. This is
Go ahead,
Suraj
please.
Bhatt. Thanks, Bridget. Thanks for quite an informative seminar. Thank you so much. I was wondering, is all the infection going through ACE2 expression all the time? You have human fibroblasts which are resistant, and then you mentioned that there is polarity in the cells, and ACE2 is expressed only on one side. Is it the apical side or the basal side?
It's the apical
but
side.
Is all the expression it's the apical side?
Yes, from the lumen. Yes.
From the lumen. I see. Yes. So in the myocytes, in the cardiac myocytes, is the infection dependent on ACE2 completely?